Everybody's got your title?
Everyone's got my title. Hello, thanks for the invitation to be here. This is great. Representing sensory reality in the brain is the topic of the symposium. But as Marie Francoise brought up earlier, let's get something straight. And that is, the symposia is really a drinking party. So really come to the after party at the end of the day and socialize, because I think that's an important part of why we're here. And then we can distort sensory reality, or representations in the brain. So I'm going to talk a little bit about how flies represent sensory reality. And you all know that sensory modalities are actually not working independently of one another, not entirely. Right? You realize, of course, that your sense of flavor is driven by the convergence of your sense of smell and your senses of taste. If I were to pass around licorice and cherry jelly beans, and you had to hold your nose and you didn't know what color they were, you wouldn't be able to, most of you would be able to tell the difference in the flavor of them. You would guess wrong. You would guess that they're sweet, but you would know if they were cherry or licorice. Likewise, you hardly recognize where I'm standing because you can see me when you're not looking at me and you're looking at my colorful flies. You can hear my voice and you recognize that there's a convergence in the spatial location of my voice and my image. You realize that, of course, when you are on a boat and you are pitching in the waves, you might get seasick in part because your vestibular system is registering one frame of reference of inertia and your visual system another, and they don't match. So processes of representing sensory reality in the brain are actually oftentimes multimodal, and we understand a lot less about how those interactions occur than we do about how the individual modalities work on their own. Flies are a great model for studying multisensory integration because they do it so well. So here's a couple of images of the fruit fly Drosophila melanogaster, one from Wikipedia and one on the right from my own lab. I spend my career gluing flies to sticks and putting them in little sensory virtual reality arenas. And I do this because I'm interested in how flies pester me on my patio. Right? It doesn't take long opening a bottle of wine on the patio of my house, the densest urban jungle on the planet, for fruit fly. And in about 15 seconds, it lands on my walnuts. And my wife asks me, where did it come from? This is unbelievable. You don't see a single fly for days. It's dry, it's arid. It's not a hospitable environment for fruit flies. Yet within seconds, one lands on the rim of my glass from presumably nowhere. And the behavioral repertoire is actually pretty simple. Flies, like most chemosensory insects, will fly upwind naturally, seeking the sources of odor emanating from their food. So they'll reflexively track upwind. It's a reflex called pneumotaxis. If they do begin to search for food odors, they might cast back and forth across the wind stream, seeking some packet of odor. If they detect a coherent chemical signature, they iron out their flight patterns and they surge upwind, navigating up that plume. And this process we might call local tracking versus a more global search strategy. And it requires spatial integration and temporal integration of the olfactory cues. But it also involves visual stability. And insects as diverse as hawk moths, mosquitoes, and flies have all been shown to demonstrate very strong visual reflexes when they're encountering an odor plume in a wind tunnel sort of experimental environment. So there seems to be some kind of integration between the visual and the olfactory system of these animals. And in fruit flies, we have this advantage in neuroscience where we have a wealth of genetic and physiological tools to be able to go in and assess the circuit components, right? At a level that Dean hopes to get to in the mechanisms of how cells and cellular networks produce a signature of time, and that is learned. We hope to be able to get to a satisfying solution for how these modalities interact at a cellular and molecular basis. So this idea of visual stability is where I'll touch upon first. What is visual stability? You all know it, but it was described formally by physicists working in biology at the Max Planck Institute in the early 1950s, where they basically decided to assume that visual reflexes and visual stability works the same essentially. The algorithms must be similar across animal taxa. So they chose a simple animal to work on. This is a beetle, and they suspended that beetle, they glued it to a stick. So I'm not as crazy as they might seem. My career choice of gluing animals to sticks. There's a rich history of this. dating back to physicists, right? So I'm on solid ground. This beetle was then handed a straw wine maze, very small, very tightly handcrafted by very skilled Germans. Teeny little wine maze made out of straw. It was so light the animal could just hold it. And it would walk on this wine maze, and it would come to these junctions in the wine maze, and it would either steer the little globe to the left or to the right. And you suspend that animal and strike a drum, and you rotate the drum around the animal, and the animal will steer to the right or steer to the left. And you can vary the parameters of the pattern, or the speed, or the intensity, the spatial and temporal properties of this visual display, and map out algorithms that produce visual stability reflexes. Now that, again, it's not as crazy as it seems. Circa 2014, there's a rodent in a striped drum. Turns out scientists have been putting animals with image-forming eyes in striped drums. Lots of animals, lots of different species of animals. So here's a crab that actually is a bat in a striped drum. And we have a zebrafish. This is more recent work looking at optokinetic stability in zebrafish. And another species, yes, the present undergraduate. So again, putting animals in striped drums is very complicated because you can quantify very rich parameter space very quickly and study how optokinetic stability or optomotor stability works. Similarly, lots of different species of animals have been used to study chemical navigation. This is an example from my lab where we basically took a tethered fly who was orienting in a circular arena that I'll show you later, exposed to an odor plume, and we prescribed some forward velocity and looked at what the flight path would be in this sort of virtual odor plume that you see in yellow. And so you can see that even in a laboratory condition, the animal sort of steers back and forth and navigates up that plume. Here's an example of a bloodhound tracking the scent of this pheasant. You can see the trajectory of this chemical navigation looks remarkably similar. And of course, the ever-present required undergraduate. In this case, this person at UC Berkeley is trying to navigate a plume of chocolate odor that had been dragged across the grass. And so the point here is that these visual, the algorithms that underlie visual stabilization and the algorithms that underlie chemical search are actually remarkably conserved across taxa. And in my lab, I wanted to understand and control both of these parameters simultaneously to look at how olfactory cues might modify or bias visual stability reflexes to enable these animals to localize chemical trails that are invisible and often turbulent. So the setup was as such. You take a fly, glue it to a steel pin, suspending that pin between two powerful magnets on a pivot so that the pin can rotate freely with essentially no inertial load on the fly that is glued to it. But it can't displace horizontally back and forth. Right? So the image of the rotating fly is captured in infrared video from below, and the system is this little magnetic tether system is surrounded by for a fly, a very high-resolution display of light-emitting diodes, LED diodes. And here's our sort of canonical striped drum. And when you look at what the fly is doing in this arena, you can see the image from below. You'll see this animal. You can see the wings beating, although it's blurry because, of course, they're using infrared video. We can rotate this striped drum around, and the fly does what every undergraduate fish, crab would do, which is rotate essentially coherently with that visual display, demonstrating this optokinetic stability reflex. It's very powerful. Then, to investigate how chemical signals might be involved in or integrated with this, we then superimposed a very low flow wind tunnel on top of this setup. So we're essentially using a multiplexer to deliver odor or water vapor, saturated vapor, through a two-channel nozzle that dribbles this fluid at an extremely low flow rate down across the fly's antennae and gets sucked into a little vacuum chamber from below. It is clear so the video can pass through it. And the reason we want this fluid to be very low flow rate is, of course, because the animals are anemotactic. If there's any wind, they'll orient right toward that nozzle. But in the absence of any attractive odor, hopefully they won't. And we can tell then, control the animal's olfactory experience, and then directly manipulate the visual experience at the same time, and tease apart these sensory modalities. This is what the data look like. Where on this axis is the animal's heading in the circular arena, right? We're at 0 degrees arbitrarily. We place the location of this small nozzle. And for these 5 different flies, individual 15-second trajectories in heading are sort of color-coded. And in black and gray, you see what these animals do when we visually place them at the start of the plume and release them when there's no odor. So they do what this fly does. They sort of meander back and forth in these saccadic trajectories, and they explore the arena. When you switch the odor on, each of these animals then remains tightly focused in the center of the arena in this long duration. Right? So they actively track the odor. During this experiment, this striped drum is stationary. The animal produces optic flow and it turns back and forth, but we're not imposing it in any way. Okay? But it itself will experience very rich visual feedback as it displaces its yaw location in the arena. And so we asked a very simple question. Do flies use visual feedback at all to navigate the position of this plume? Right? So the test is very simple, and it's illustrated by this video, which is a montage. It's not to scale. Here you'll see a live camera image of the LED display. It looks pixelated and coarse, but flies have terrible spatial vision, so to flies is like high-res IMAX. And you'll see this very blurry infrared image of the fly, and I've just cartooned the location of the odor plume. We do a very simple experiment. We take this hungry fly, and we visually lock her into the location of the plume. She's hungry. This is a plume of apple cider vinegar, highly attractive to flies. We can use red wine odor. We could use scotch, on Friday afternoons, and they love that. And coffee on Monday mornings, they'll track that. I mean, any appetitive food odor that draws them into your patio will draw their attention in this arena. And then halfway through the trial, we just do something cruel. We just dim the contrast of the display from this very high contrast black and white striped drum to an equiluminant, uniform arena. We simply ask, how does the fly react to that? And so at the start of the experiment, you see these small saccadic turns that can orient the animal back and forth across the plume, but she never leaves this location. She doesn't orient 180 degrees away like she would if there's no odor present. There's so much cool behavior going on in this video, like I could talk about for hours, but like I said, it's symposium. Later, symposium. And then here we go. Here's the uniform grayscale. It's just one fly, and she's 90 degrees away. She seems like she might get a notion at some points and hit the plume, but statistically, this behavior is essentially indistinguishable from when there's no odor present at all. She's completely out of the plume within a few seconds. And data that just demonstrate that are illustrated here, right? So here's just 5 individual flies that are with a paired design, so we see that the individual trajectories are color-coded. And we start each of these animals at the same location in the arena. We visually clamp them at this location, or aiming right at the odor source. And these hungry flies, if they're presented with this high-contrast stationary visual display, they track the odor beautifully. If, on the other hand, we switch the visual panorama to this uniform, untextured display, they steer out of the plume in a manner that's statistically indistinguishable from when there's no odor present at all. That's very strange, right? This is a fruit fly. Its job in life from the time it is born as an adult is to disperse from where it was born, find the source of some attractive odor, where it's going to find mates and lay eggs and reproduce, and it should just be able to close its eyes and locate the food or source, but it's unable to do so. And so the question is, what is the mechanism underlying this peculiar interdependence between vision and olfaction in these animals? Can we get to it at a synaptic or cellular level analysis? So to test the idea, the first hypothesis is very simple. Somehow, odor must modulate optokinetic or optomotor reflexes. It's the simplest hypothesis to test. Does odor somehow modulate or change the gain of visual stability reflexes? reflexes in the fly. To test this idea, we flip to another behavioral paradigm where now the fly is not free to steer around in the plume, but rather is fixed rigidly, and we track the animal's attempts to steer using an analyzer that traces the shadow of, or changes in the shadows of the beating wings cast by infrared light onto the sensor. So when the fly tries to steer left, the shadows are displaced in one direction, and if the fly tries to steer right, the shadows are displaced in the other direction, and we can couple that time-varying signal to the dynamical rotation of the pattern on the LED display. And so in this closed-loop feedback system, for example, we know the fly is highly engaged in tracking behavior, for example, by presenting the animal with a single solid vertical stripe. And all of the motion that you see in these stimuli in the movies are being directed by the fly itself. We're not imposing any of these visual dynamics. You can see the wings beating back and forth, and they're sort of changing amplitude, and that's directly in sync with the dynamics of the visual display. So the animal can control its own sensory experience, and we use this as a proxy for understanding how engaged it is in these visual tasks, which I won't talk about, but just to give you an indication of how strong this is, here would be the animal's heading over time with respect to the location of this vertical landmark. It's like a piece, it's like a tree or a stalk of grass that the animals naturally navigate around or land on. And you can see that basically this single fly keeps that stripe oriented right in its frontal field of view. And some data that Karen in the lab just collected a few weeks ago just illustrates this very powerfully, that the probability distribution of the location of that stripe under closed-loop control is very tightly fixed at 0. The question is, how are these visual responses enhanced or changed in some way by the presence of an attractive odor? And so we just equipped the arena with the same kind of nozzle suction device, essentially, as you saw in the previous hearing, where we can just flip on and off an odor source that is blown at a very low flow rate over the fly, sucked away by a nozzle from beneath. And then we do 2 different experiments. On the left, it's very simple. We just do what people have been doing with striped drums for generations, and that is oscillate this drum back and forth at different speeds, and then we record the animal's steering response, which is a proxy for how it's trying to stabilize that image motion. And you can see in the absence of odor, in blue, you have some amplitude spectrum of the average steering response, and that when the same animals are given, the odor is switched on, you can see the amplitude goes up. So the gain of this input-output transformation of a very, arguably one of the simplest reflexes, optokinetic stability, ever studied, the gain of that reflex goes up in the presence of odor. There's another method that we use to test that. It's not particularly germane to the discussion here, but we can use more of a white noise stimulus and record essentially the impulse response to these perturbations, and you see the same result, that when there's an odor present, the strength of the steering impulse response to this white noise sequence is larger. So odor increases the gain of optomotor stability responses. And Karen has also then recently shown that if the animal is actively controlling the orientation with regard to this visual landmark, then the orientation histogram is even stronger in the presence of odor. So very clearly, the olfactory signals are modulating visual behavior in a robust and repeatable way. And so the conclusion of this, right, is that olfactory signals, and this is a strange mix of modalities, right? The examples I gave you, taste and smell, these are chemical modalities and are processed by similar kinds of circuitry in the brain. The classic one, the visual auditory integration, is processed by tissue maps that are actually in register. Right? Spatial, and they're laid out, a spatial map of the world. Chemicals are not, there's no spatial spectrum to chemicals. Yet here we have this visual sense, and this olfactory, this chemosensory sense, somehow coming together, modulating one another. How does this work? In flies, we have a hope of addressing this. And the tools that are available are actually very powerful. But even before that, the structure of the visual system in flies is actually not that unlike what you would see in the sort of canonical vertebrate. And that is that there's layered processing stages in the visual pathway, and that's indicated between these 2 graphs. This is a review that Josh Staines and Larry Zapersky did a few years ago. The color code of the different cells is only meant to indicate that there are rough analogs if you will, or parallels in the types of cells represented in the columnar organization of the visual pathway. There are processing stages in the LGN, the tectum and colliculus, show properties, circuit properties that are very closely parallel. The processing going on in the fly, lobula, and lobular plate, and the visual cortex and the fly protocerebrum have these very high order processes that are much less well understood, but roughly show similar kinds of parallels at multiple levels of analysis. The fly brain is represented here on the right. This is the right optic lobe. These are the three dominant neuropil. The medulla, which is represented here, this layered structure, columnar inputs, the lobula and the lobular plate. And we have genetic reagents to label reproducibly individual cells and kinds of cells within the visual system, for example. So what you're seeing in green is a single, it's two different cells. These are columnar cells, T4 and T5. Each and every one of them is labeled with GFP, but nothing else in the brain lights up. There's 100,000 neurons here, and you're looking at 100 of them, roughly. Okay, labeled by this GAL4 graph. How does vision work in flies? Well, here's a sort of simple breakdown. Essentially what's happening is that there's a lot of processing going on in the medulla. All these columnar elements converge onto this T4, T5 pathway. Each cell in here is computing motion over a very small region of visual space. Right? So you have a purple box here representing essentially a single columnar input between the medulla and this region called the lobular plate. And this purple box is computing motion over a very small region of visual space, and the retinotopic organization of those local motion inputs is collated or summed by a downstream wide field integrating cell that resides here in the lobular plate. The synaptic architecture of how this works is actually really well worked out. From here to here, from this part to this part, it's less well worked out, but we do know that integration of these local motion inputs by these wide field cells underlies optomotor stability behavior, for sure, no question. The cells that reside there are actually sort of classically defined. And so the inputs to this entire pathway, these local motion inputs, in green here, this T4, T5 pathway, we hypothesized if motion vision is so important to olfactory navigation, then silencing these local motion inputs ought to have a profound influence over the animal's ability to track a plume of vinegar in our arenas. And so in this axis, you're looking again at this magnetic tether system where the odor is placed here, and for three different representative animals in three different genetic conditions. The two controls where we're using a UAS to drive an inward rectifying potassium channel in the cells labeled in green, and nowhere else. These two are genetic controls, and you see that those two animals essentially track the plume just fine. The experimental, where here is being expressed in these green cells, the T4, T5 pathway. The animals steer around in the arena as if there's no odor at all. And you see that essentially borne out in the compiled data where the mean time in plume is plotted here. For the experimental group, between no odor and odor, there's no difference. The animals are not tracking the plume or spending more time in it by comparison to the water control. Yet the genetic controls do track the plume significantly well. So here we now know that this integration step between olfaction and vision is required in these behaviors that we've already shown. The optomotor behavior relies on this motion vision circuit, right? And so this motion vision circuit feeds this region called the lobular plate. So the question is, well, you know, are postsynaptic targets of these local motion computations involved in this integration step? The postsynaptic targets have been known since 1960, and they are beautiful cells. They were identified by the German groups that I showed earlier, the same group that worked on the beetle, then began doing sharp electrode recordings from these cells in the lobular plate. This is a different view of the lobular plate. This is sort of a plate view of that layered structure. And what you see there are these gigantic dendritic arbors. Each one of these spans roughly 150 degrees of visual space. And this one cell, HSE, is the postsynaptic target of hundreds of the individual columnar T4, T5 local motion inputs. This wide field neuron has very standard characteristics. First of all, it's directionally selective for horizontal motion. So if you rotate a grating up and down, back and forth, down, in all these different orientations, what you see is the directional preference for horizontal motion in one direction, inhibited by motion in the opposite direction. So this very tight directional tuning, canonical demonstration of one of these pre-motor integrated, visual integrated cells. Another feature is that the cell is tuned to the width of the stripes, if you will, the temporal frequency of the stripes as they pass a certain point in space. This would be easily transformed to a velocity axis if you kept the stripe pattern the same and just rotated it faster or slower. So there's this tuning function to essentially speed, and there's a directional selectivity. We found a cell in the lobular plate of flies that hadn't been described before, and it's, again, one of these giant tangential neurons that has this beautiful, gigantic dendritic arbor and sends its axon into the premotor areas of the central brain to influence optomotor behavior. And the question then is, of course, is this one of these canonical wide field motion integrating cells? Here's calcium imaging, 2-photon calcium imaging of one of the a big slice of the dendrites, and you can see that it's highly directionally selective. The dendrites are excited, the calcium flow is excited by motion in one direction, but not the other direction. It's a fairly diffuse looking excitation because, of course, you have to realize that at each point in the dendritic arbor, it's receiving local motion input, but of course all those currents are being summed here. So if you switch the recording to the region where all those currents are already being summed, then what you get is an extremely strong signal-to-noise ratio response to motion in the preferred direction. So when you turn the visual display on in this preparation where we have this fly under a 2-photon imaging microscope, you get very strong excitation. There actually are error bars there, but they're very, very small. So it's directionally tuned. You can demonstrate that by orienting the grating in different orientations, and you see this really nice directional tuning profile. So this cell produced back to front motion across the eye, posterior to anterior, and is inhibited by front to back motion. And its temporal frequency optimum, or sort of velocity tuning, if you will, is exactly the same as in that other canonical HS neuron. We call this cell HX because it is a horizontally sensitive cell. And the question was then, is this HX neuron sensitive to input from odor? The experiment's very simple. The animals, in this case, are actually non-behaving, right? They're just fixed to the stage, looking down at this LED screen. It's exactly the same as the one we use in the behavior, and we equip that with an odor plume essentially identical to the ones we saw in the behavior. And the question is, are the visual dynamics, is the strength of the visual response in these cells modulated by odor? And you can see roughly what the experiment looks like here. Now we've switched the field of view to this integrated region in the dendrite. And in the second, it's kind of hard to see the graining reproduction here, is that in the second motion pulse is paired with odor. So the response is larger when the odor is turned on, and it remains a bit higher than it was for the first motion stimulus. And we can take a look at what those data are, and it shows that yes, the visual response is very strong, but it is modulated subtly, yet significantly, by the paired presentation of odor on the antennae, the chemosensory antennae. In this case, that odor stimulus far outlasts, or the effect of the odor far outlasts the acute stimulus. I'm not going to get back to how we think that operates in a minute. But the point is that if you look at this response, you compare these naive to the odor-paired trial across multiple animals, you always get the effect. And if you turn on water vapor, of course, you do not.
Right. Is the effect directions sensitive?
Yes, we've never looked at whether the olfactory modulation occurs to the null, the anti-preferred inhibitory direction. It's an interesting idea. Nobody really has a satisfying description of what the inhibition is good for. We actually never know the cells that produce it, but that inhibition hasn't been, because there are cells that have, this cell, the HX cell is sitting in the next layer from the HS cell. They have a directly opposed preferred direction. The way that the outputs are presumed to be wired onto the premotor network. There is nobody has posited a satisfying need for the inhibition. Rather, it's thought that the inhibition is coming from the fact that coming from the way that the directional selectivity is shaped. Right? So it's essentially a byproduct of the strong directional selectivity. Now the question is, look, fine, you've turned it. This prep is glued to a stage. It's there's like a third of a tiny microscope glued to its head. It's cold. It's in a miserable state. And you blow this attractive odor across the antennae, and the whole fly gets aroused. Like, I have a chance. It smells good. What are you doing to me? And maybe the whole brain is aroused. Right? That's an important control to be able to distinguish. So we recorded from these presynaptic inputs I told you about, these local motion detecting inputs, T4, T5. We also recorded from the neighboring HS neuron, which is sitting in the exact same nerve pill, one layer over, receiving exactly the same kinds of inputs from the T4, T5, just different directions. And you don't see, in either of these cases, any modulation by the presentation of odor. So there's no way that there's this global arousal effect in the nerve pill. It's not coming, and further, it helps us identify the fact that the effect of odor must be local to the lobular plate, not presynaptic coming in from the medulla or upstream regions. Right? So odor doesn't have an effect on the local motion inputs to this wide field integrating premotor neuron, nor does it have, as far as we can tell, any impact on the operational gain of a neighboring sister wide field motion integrating neuron. So we get to this point in the sort of trajectory of research in the lab, and I'm ecstatic. Right? I'm like, okay, this is great. I've been studying this general phenomenon of olfactory influence optomotor gain for years. Now we've got a cellular sort of correlate to that. And, I'm ready to go. I think this is terrific. Let's try to publish this. And of course, Sarah, the postdoc, says, well, no one's going to get as excited as you. I think the quote was like, nobody's ever as excited as you. We can drill down deeper, right? So, of course, we can, right? So the question is now, well, what's the source of the modulation? And we've localized it to this region of the brain that contains about 60 maybe 70 different kinds of these wide field integrating neurons. But that's it. There's some local stuff that wires back and forth. But the dominant architecture of this neuropil are these gigantic, beautifully wide field motion integrating neurons. And so recently, a paper came out of Michael Dickinson's lab that showed that a octopaminergic system. So octopamine is the neuromodulator is the equivalent of norepinephrine in flies. Okay? This is the juice. This is the fly, the excitation hormone, right? Or neuromodulator. And there are octopaminergic neurons that innervate a vast area in the brain, but here's the optic lobe, the right optic lobe. And blue is just a synaptic marker identical to the purple that you saw earlier, showing you what the tissue looks like. And in green, you're looking at a GAL4 that's labeled by TDC2. So TDC2 is the gene that produces the enzymatic precursor for octopamine synthesis. So these cells, in green, release octopamine into the visual system. And what Marie Suber had shown was that, that these cells are important for modulating, in a different context, the gain of these wide field integrated neurons that I just showed you. So of course the question is, here's the lobular plate, TDC2 expression, here's the lobular plate, HX neuron. Are these two talking to each other? Is octopamine, which could be used as a neuromodulator, excited by the olfactory modality to influence the visual modality? So we have some reagents to test that. So the first thing we did, in collaboration with Camilla Larson in London, was to use a really clever technique available in flies and mice actually as well, GFP reconstitution across synaptic arteries. And the idea is that you use one GAL4 driver to express part of the GFP, and another GAL4 driver to express another part of GFP. And then only where those two cell types are in close apposition is GFP, the full protein, reconstituted. Only when membrane touches membrane in cells where you put one half of the GFP expressing gene. My laser pointer seems to be up. Hang on a second. See, I'm so excited I unscrewed the barrel of the laser pointer. So one half of the TDC2 expressing GAL4 is being, one half of GFP is being expressed by the octopamine GAL4 that you saw on the previous slide. The other half is being expressed by the HX GAL4 that you see here, and you bring them together, and what you see then in the result is that these little puncta, these green puncta, exist where the two intersect. And all these green puncta reside in the lobule plate, in exactly the same profile occupied by the dendrites of the HX neuron. So that's evidence that suggests that perhaps the HX is either in very close apposition to the membrane of this octopamine system, or it forms active synapses. Okay, we don't know the answer. Another important experiment was to see, well, is the octopamine circuit driven by odor? Where is the signal coming from? Chemosensory signal. And so we tested, by expressing GCAM, of course, in the TDC2 circuits, and then pumping odor out. We saw that, yes, indeed, this octopaminergic pathway is activated by odor. And then finally, what if we removed the capability of the octopaminergic system to function properly? And we collaborated with David Krantz to generate a fly that is mutant for the vesicular monoamine transporter that the octopamine circuit needs to dump the neuromodulator. So there's no synaptic transmission, essentially, of octopamine in these animals. We then have to rescue that with either a mutant form of a rescue construct. Those animals do not track in the plume. Or we rescue with the native active form of the monoamine transporter. Those animals do now track in the plume. And of course, those animals tested in water do not track in the plume. So in an animal that has essentially zero vesicular monoamine transport capability in the brain, we've rescued it only in this octopaminergic circuit, and that alone is at least sufficient to rescue olfactory tracking behavior. Right? Again, suggesting that we've sort of closed the loop here. So it's very satisfying, right? We've now got a cellular and we think even a molecular component that explains how these two modalities are wired together. And the question that Sarah then took off with was, well, is it just odor? Is there other state-dependent changes that the animal might experience? Rather than it's flying around, it experiences some olfactory signal, and that olfactory signal is enhancing release of octopamine locally in the visual pathway, which turns up the gain of this visual input to motor output stability system, and the animal actually rejects visual motion more powerfully and flies straight up the plume. But other states might influence visual processing as well through the same kind of system. So one state that's important to flies, not just food odor, but starvation and dehydration. These animals are starved and dehydrated. They get agitated. They behave in ways that they don't behave when they're fully sated. So she did a very simple experiment where she compared fed and hydrated animals flying in this little virtual olfactory plume. And of course, when you put the odor on, they track in the plume. When there's water vapor, they really don't. Then you take those animals and you dehydrate them. And now the dehydrated flies track the water plume as well as they track the food odor plume. They don't track air, which you never thought is a reasonable control before. That's the only control we have left. Okay? So starting and dehydrating these animals now has them actively tracking the water plume, water vapor plume, as well as they would track the food odor, and when they're sated, they don't do that. Right? And the question is, okay, well, what happens to the optomotor behavior? Same thing. When they're fed and hydrated, you present an odor pulse, and the odor increases the gain of this optomotor response. And starved and dehydrated animals, you test them with water and odor, it doesn't make any difference. You have the full gain in optomotor response already. So starving and dehydrated just pegs the system, if you will. When you image from this HX neuron, you get essentially the same result. In the dark gray here, you see a repeat of the experiment we already saw, where the motion-dependent response goes up where there's an odor pulse, but the entire, in light gray, is then shown what happens when you starve and dehydrate the animals. The gain of this entire system is just pegged all the way up at maximum. And so she's now teasing apart, trying to tease apart the neuromodulatory mechanisms for how that occurs. And we think it involves dopaminergic signal as well as dopaminergic signal. So, just to summarize then, you've got this idea is that local motion computations occur in the earlier in the visual pathway, locally over small regions of the visual space, and you assemble all of those into a summation that's integrated by these huge wide field motion integrating neurons in the lobular plate, which then drives optomotor stability, right? It provides a feedback mechanism for the inputs to the pathway. When there's an odor then in the system, that results in octopamine release, which is then impinging upon this integration step very specifically to turn up the gain of the motion signal at the lobular plate tangential cells, these integration neurons, and enhancing the optomotor stability. Now, an interesting corollary, of course, is that this is the way the system operates when there's an attractive odor or if the animals are dehydrated, but when they're sated and there's no odor, it operates like that. So the increase in gain makes sense to me. The decrease in gain, I think, is just as interesting. Why are they operating at sort of this suboptimal level where the optomotor gain is lower and the response magnitude of these cells is lower? But we'll save that for discussion. So the general idea here is animals fly upwind, they cast around, and once they hit an odor source, the local plume tracking strategy relies on the fact that odor activates neuromodulatory circuitry, octopamine, to enhance response gain of motion vision pathways and then strengthen optomotor stability behavior. And very importantly, when the odor signal is lost, the gain of that system turns back down. Okay, so just to quickly thank a few people. Of course, Sarah Wasserman is a postdoc in the lab that did most of the work that you see here, at least all the recent work, the recent behavior, all the imaging, and some of the work before that came from some of the former grad students in the lab. Karen provided some terrific early pilot data. Other folks in the lab, Mehmet, Jean-Michel, and Patrick, are not involved in this project, but of course are tangentially related today. Thank you for your time very much.
Questions? You're all stunned.
Yeah, it's okay. He's done. I'm just stunned. Alan, yeah.
There's another way of expressing this motivation, the effect of the factor stimulus. In that case, the effect of that mainly on the motor response to a visual input. You have several predictions. One, that if you had a really attractive odor, there would be a bigger magnification factor. And maybe the neurons you're looking at are in fact motor or premotor neurons. In any case, the importance of the olfactory stimulus is to make the fly want to continue to keep going in the direction of the odor. And it's not visual cues that tell us what direction it's coming from when they move, it moves. I don't know whether you didn't use the word motivation. Is that?
I don't use the word motivation only because I think it's difficult to define. I have no doubt that what you're seeing is a instantiation of motivation. Right? Certainly that's true. You know, flies are starved, they're dehydrated, they're hungry, they smell something good, they are motivated to track that plume. I think that this sort of cognitive, definition of motivation is difficult to grapple with, but this isn't. So I don't mind if you consider this to be like an instantiation of motivation. However, I don't use the word motor. Did you say motor search? No, motivation. You're just talking about motivation. There's no doubt, that this is, but what is motivation? I mean, yes, your motivation and the result of what you do in a grocery store is strongly dependent on your hunger state when you walk into it. Right? I don't send my wife to the grocery store hungry. She comes back with Diet Coke potato chips. Right? It's really right. I mean, and she goes, "What did I do?" She even joked with this, but it's
the first state at which you see a change in the response to the vision stimulus is how many neurons there?
This is, I'd say, probably within 6 synapses of the photoreceptor. Probably even less than that. The effect in this wide field motion integrating neuron, this is considered rather a peripheral sensory. Is that me? I don't even have a microphone on. I'm feeding back to that microphone. Too much energy. So, I don't, this is considered a peripheral sensory circuit. Now listen, it turns out that in zebrafish retina, odor presented in the animal increases the gain of off ganglion cells, or off bipolar cells, what are the other, I can't remember, but not the on cells, in the retina, right? So I think that the lesson here is that the idea of motivation can be probably mapped to very peripheral, you might call low-level processing events. It doesn't have to be a very high-order cognitive process. It may occur at higher order. There's certainly higher-order structures in the fly that are involved in long-distance navigation, for example, or courtship behavior and motivation for courtship behavior and such. And those regions of the brain may be involved in this as well, but we're at least demonstrating that part of this is occurring at a very low level. Now, the other question you mentioned is actually kind of interesting. So, since nobody asked the question, does anybody else have a question? Otherwise, I'm just going to tell you something you should ask. Felix, dang it! Are you asking? Go ahead. Yes. When you turn on it, Sarah has a whole paper. So when you turn on an aversive odor, it's great. For years I gave this talk. I'm only working with attractive odors because that's what flies care about. They're seeking active plumes, and it's easy to test that. People always say, "Well, you tested aversive odors." I said, "Well, it's not the logical inverse of attraction." And I expected in our reading, when we turned on an aversive odor, benzaldehyde, turns out for reasons totally mystifying to me, it's the smell of almonds, is totally aversive to fruit flies. When you turn on a plume of benzaldehyde in this little ring, which seems to have a very narrow plume, we measured it, and the flies tell us that it looks like a very narrow plume, that if you turn on an aversive odor, they'll just steer around the arena, and they'll just avoid that one little spot, right? And so, what do you learn from that? That's not what they do. You turn on a narrow plume of aversive odor. They turn and they actively orient with as strong accuracy in the exact opposite direction. You would very well, you absolutely would. There are food odors that are so unbelievably repellent. There's a mold, for example. The mold that grows in the oranges after a while. Extraordinarily, probably the singular most aversive chemical compound ever isolated in the history of humanity. For animals, for any animal. Okay, it's this mold. Unfortunately, it's extraordinarily toxic to flies and probably you too, so you don't want to cook that stuff. But the aversive odor, the point is the aversive odor provokes exactly the same tracking algorithm, and it turns out that we think operates similarly on the optomotor pathway in the visual system. That the algorithm is exactly the same, it's just that the animal flips the sign of the tracking response. So instead of always tracking toward the gradient or toward the odor, it tracks exactly away from the odor. But the visual stability is engaged in just the opposite direction. So,
Thanks, Mark. We're going to go back to a break for a couple of minutes.